Have you heard about membrane fluidity? It refers to the viscosity of the lipid bilayer of a cell membrane or a synthetic lipid membrane. Lipid packing can influence the fluidity of the membrane. Viscosity of the membrane can affect the rotation and diffusion of proteins and other biomolecules within the membrane, thereby affecting the functions of these things. Now, a lot of different factors can affect membrane fluidity, but the three most important ones that we are going to discuss today are first the temperature, second the cholesterol, and the third, which is whether we have unsaturated or saturated fatty acids.
Remember that phospholipids makes up the cell membrane. Each phospholipid consists of a phosphate head and links to two fatty acid chains that creates two layers of cell's double membrane. Now let's find out how temperature became a crucial factor that affects the fluidity of the membrane. When phospholipids are in low temperature, their mobility is decreased. That's why they are huddled together, resulting for the fluidity of the membrane to decrease. This is known as the crystallized state. Thus, as the temperature decreases, the fluidity of the cell membrane also decreases. Now, what about at high temperature? At high temperature, the phospholipids have a little bit of energy, causing it to move and have distance from each other. Therefore, as the temperature increases, the fluidity of the membrane also increases. Now, the key idea of this, the fluidity of the membrane is proportional to the state of phospholipids in a particular temperature.
Another factor that affects membrane fluidity is cholesterol. So cholesterol lies alongside the phospholipids in the membrane. There is more cholesterol in some membranes and less in others. Cholesterol has different effects on membrane fluidity at different temperatures, and we will discuss more about that. At low temperatures, phospholipids still tend to cluster closely together. This cholesterol between them is going to push them apart and say, "Hey, move more, you're too tight, I need some distance." And in the context of high temperatures, as the distance between phospholipids increases, the fluidity can also increase. At high temperatures, phospholipids are already far apart. The cholesterol in between them will cause the phospholipids to pull themselves closer together because they want to attach to it and say, "Hey, I like the cholesterol, I want to get closer to it." So now, there's more stuff inserted throughout the membrane, and so the molecules in the membrane are now closer together. As a result, the fluidity decreases. Cholesterol is really interesting because at low temperatures, the fluidity will increase. At high temperatures, the fluidity will decrease. Thus, cholesterol functions as a buffer, preventing lower temperatures from inhibiting fluidity and preventing higher temperatures from increasing fluidity too much.
Now the third factors are saturated versus unsaturated. We will find out how these two fatty acids affect membrane fluidity. Comparing the presence of saturated and unsaturated fats, we are talking about the fatty acids chain that are hanging below our phosphate group. Saturated is represented like straight line where every point is carbon. So the question is, how will this affect membrane fluidity? Since saturated fatty acids are straight lines, and we are trying to represent the fact that these straight lines stack together really well. They stack pretty closely together. And since the distance is pretty small, our fluidity will be affected and expected to be low.
Now let's talk about unsaturated. Unsaturated, on the other hand, its structure is pretty different from saturated, as you can see kanina, because it is like zigzag and sometimes it has double bonds. So how will this interact with multiple fatty acids next to them? Contrary to saturated where it stuck neatly, in unsaturated it doesn't because of its structure that has bend. Because of this bend, it affects the fluidity. In the image, as you can see, phospholipid has bend in it, and it represents the unsaturated fatty acid. And because of these unsaturated bends of phospholipid, it resulted to have more distance affecting its fluidity to be high. So since there is more distance of phospholipids, the fluidity increases.
In conclusion, these three factors, the temperature, cholesterol, and unsaturated fatty acids, really influenced the fluidity of membranes. In temperature, it has huge effect in fluidity because as the temperature increases, the membrane fluidity also increases. In cholesterol, where it acts like a buffer, it inserts itself and does something unique that affects the distance of phospholipids. And we all know that the distance has also effect on membrane fluidity. In unsaturated fatty acids, because of its structure, it affects also the distance, resulting to high fluidity of membrane. Although the lipid bilayer structure is quite stable, its individual phospholipid and esterol molecules have much freedom of motion, which is the main reason of fluidity is property of bilayer.